Tes, 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 tes. Good morning, ladies and gentlemen. Welcome to the International Webinar of Pharmacy Study Program of Health Polytechnic of Pangkalpinang. My name is Rati Puspita Kusumadewi Purba, who will host our webinar today. It is our privilege to have you with us today. We are now ready to begin the webinar. Make yourself comfortable, and I hope you have read the rules mentioned on the previous slide. Thank you for your kind cooperation and attention. Bismillahirrahmanirrahim. To our highly respected, the speaker who have been present with us, Associate Professor Dr. Mogana Sundari Rajagopal from UCSI University Malaysia, respected Director of Health Polytechnic of Pangkalpinang, Mr. Ahyat SKM MSI, respected Vice Director 1, 2, and 3 in Health Polytechnic of Pangkalpinang, respected Head of Academic Subdivision, Head of General Administration Subdivision, and all level of management in Health Polytechnic of Pangkalpinang. Respected lecturers and staff of the Pharmacy Study Program and all distinguished participants. Assalamualaikum warahmatullahi wabarakatuh. Alhamdulillahirabbil alamin, all praises to Allah Subhanahu wa Ta'ala, who always gives health and blessings to us, and also the opportunity until now, so that we can gather virtually in order to attend this webinar. All attendees, we welcome you to the International Webinar of Pharmacy Study Program of Health Polytechnic of Pangkalpinang with the theme, Exploration of Medicinal Plants as Anti-Cancer Candidates. I would like to extend a very warm welcome to all of you. We appreciate you taking time out of your busy schedule to join us today. In this webinar, we gather many participants who came from various cities, both in Indonesia and abroad, also came from diverse background and profession. All the respected audience, the overall event that we will run on this webinar is as follows. First, opening. Second, sing Indonesia National Anthem. Third, welcome speech from the Head of Pharmacy Study Program of Health Polytechnic of Pangkalpinang. Fourth, opening speech followed by the opening of the event officially by the Director of Health Polytechnic of Pangkalpinang. Fifth, group photo session. Sixth, main session of international webinar with the theme Exploration of Medicinal Plants as Anti-Cancer Candidates. And seventh, closing. Ladies and gentlemen, let us begin this event by saying lafaz basmalah together. Bismillahirrahmanirrahim. Sing Indonesia National Anthem, Indonesia Raya. Ladies and gentlemen, let us listen together, Indonesia Raya. The next event was a speech from the head of Pharmacy Study Program. Ladies and gentlemen, let's listen together to the welcome speech from the head of Pharmacy Study Program. Please welcome Mrs. Eva Dewi Rasmawati Purba, MKes.
Thank you, Mrs. Rati, for inviting me to give a welcome speech. Good morning. Best wishes to all of us. Respected the speaker who have been present with us, Dr. Morgana Sundari Rajagopal from UCC University Malaysia, Director of Health Polytechnic of Pangkalpinang, Mr. Ahyat SKM MSI, Vice Director all the heads of program study, lectures, and all distinguished participants. Let's thank God for His grace so that we can carry out international webinar activities on the topic exploring medicinal plants as anti-cancer candidates. We would like to welcome our speaker, Dr. Morgana Sundari Rajagopal from UCCI University Malaysia, and all of the webinar participants who attended this morning. This is the first international webinar we organize at LRGH in the study program, and it is an honor to have Dr. Morgana here as our guest speaker, and we look forward to continue collaboration. On this occasion, the webinar focused on exploring medicinal plants as anti-cancer candidates. As we know that there are many ways and methods of cancer treatment, such as chemotherapy, radiotherapy, surgery, and so on, but some cause side effects. Until now, there are still many people choosing alternative cancer treatment using medicinal plants or herbs. The Bangka Belitung Islands still have a well of medicinal plants or herbs that are passed down from generation to generation and have even been claimed to prevent or treat cancer. For this webinar, is considered very important in increasing knowledge and understanding of the compounds contained in medicinal plants as potential anti-cancer agents. We hope that this webinar will provide the benefits for all participants. At the end of the speech, let me chant a pantun in bahasa. Mengorak langkah, musafir berkelana, sambil mencari bunga keladi. Acara webinar marilah simak bersama sebagai bekalan di kemudian hari. That is all, and thank you very much.
Thanks to Mrs. Eva for the welcome speech. The fourth event is the opening speech will be followed by the official opening of the webinar. Ladies and gentlemen, please welcome Director of Health Polytechnic of Pangkalpinang, Mr. Ahyat SKM MSI.
Bismillahirrahmanirrahim. Respected the speakers who have been present with us. Dr. Morgana Sundari Rajagopal from UCSI University, Malaysia. Vice Director 1, 2, and 3 in Health Polytechnic of Pangkalpinang. All the heads of program study, lecturers, and all distinguished participants. Assalamualaikum warahmatullahi wabarakatuh. Best wishes to all of us. Let us give thanks to Allah Subhanahu wa Ta'ala for the blessings and guidance given to us, so that we can take part in the international webinar with the theme Exploration of Medicine Plants as Anti-Cancer Candidates. Thank you and welcome to key speaker, Dr. Mogana Sundari Rajagopal from UCSI University, Asia, and webinar participants who attended this morning. The high cases of cancer can certainly be our initial vigilance to one, minimize the factors related with this cancer. There are several things that they are then related to incidence of cancer. According to the Indonesian Cancer Foundation, one of the causes of high cancer cases in Indonesia is environmental conditions that continue to produce carcinogens such as cigarettes, processed meats, and others. Other causes that also affect, such as the habit of staying up late, lack of exercise, and eating too much. In development, the treatment of cancer is done with chemotherapy, radiotherapy, and surgery. Some of the most commonly used chemotherapy drugs are anti-metabolism, DNA interactive compounds, antitubulin compounds, hormones, and molecular targeting compounds. However, the use of chemotherapy drugs can cause side effects such as hair loss, bone marrow suppression, drug resistance, gastrointestinal lesions, neurological dysfunction, and cardiac toxicity. As one of the efforts of treating cancer, the use of medical plants or herbs is an alternative chosen by the community. In development, a lot of research has been carried out in the field of natural ingredients for the discovery of new cancer drugs. At present, various herbal plants and single active compound isolated from herbal plants have been found, which can be used as anti-cancer agents. Therefore, this webinar is important to carry out. One of the objectives of which is to develop insight and increase understanding of student participants potential of medicinal plants as anti-cancer. I hope that the participants can take part in this webinar as well as possible, so they can get something that will be useful later. By saying Bismillahirrahmanirrahim, I officially open the international webinar with the theme Exploration of Medicinal Plants as Anti-Cancer Candidates. That is all, and thank you. Billahi taufiq wal hidayah. Assalamualaikum warahmatullahi wabarakatuh. Thank you.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much, sir. It was enlightening speech. With a speech from our director, then the whole agenda for today officially started. Before we enter the main session on today's webinar, let's take a group photo. The event committee, please prepare. Okay. The first slide. 1, 2, 3. Next. The second slide. Okay, don't forget to smile, ladies and gentlemen. Okay, ready for the second slide? Okay, the second slide. 1, 2, 3. Okay, next. The third slide. Ready? Okay, 1, 2, 3. Okay, and the fourth slide. Okay, fourth slide. 1, 2, 3. Okay, thank you very much. Dear ladies and gentlemen, let us join together the main session of our event today, International Webinar on the Pharmacy Study Program of Health Polytechnic of Pangkalpinang with the theme Exploration of Medicinal Plants as Anti-Cancer Candidates. Have attended with us speaker at this webinar, Associate Professor Dr. Moganda Sundari Raja Gopal from UCSI University, Malaysia. The main session will be guided by our moderator, Ms. Zahria M. Pham. But before we call our moderator to lead the main session, let me read her CV. Our moderator is Ms. Zahria M. Pham. She has got her Bachelor of Pharmacy at Institute Science and Technology Nasional, Jakarta, and Master of Pharmacy in Pharmacology and Toxicology at Universitas Indonesia. Some of the researches she has done are as follows. 1. Evolution of co-administration of roselle water extract, hibiscus subdarifael, and aspirin for antiplatelet therapy in male Spritz dolly rats. 2. Pattern of using herbal medicine as complementary therapy in hypercholesterol patients at Pangkalpinang City Health Centers in 2020. 3. Ethnopharmacology study of medicinal plant in traditional herbal medicine in Bangka Barat, Bangka Belitung. And she is a lecturer in pharmacy study program of Health Polytechnic of Pangkalpinang until now. Thus, I have read the CV from our moderator today. Ladies and gentlemen, without further ado, now please help me welcome our moderator, Ms. Zahria M. Pham. Floor is yours.
Okay, thank you very much, Mrs. Ratih Puspita, for inviting me to guide our webinar today. First, I want to greet our speaker. Hello, Dr. Mogana. Hello, Dr. Mogana, are you with us?
Hi, good morning, Dr. Zaria. Can you hear me?
Hello, okay.
Can you hear me?
Okay, thank you, Dr. Mogana. Thank you for your coming for our webinar today. Ladies and gentlemen, the webinar is about to begin. All participants, please attention. Your device must be on mute mode. All right. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everybody. First of all, I would like to thank for attending to this webinar, and I hope a lot of useful information can be obtained from today's webinar. Ladies and gentlemen, I am Zahria Supardi, moderator of today's webinar. I'm very happy to see you in our meeting Zooms. Welcome everyone to today's webinar. The topic of our webinar today is explorations of medicinal plants as anti-cancer candidate. Ladies and gentlemen, our speaker is Associate Professor Dr. Mogana Sundari Raja Gopal. She is a dean of the Faculty of Pharmaceutical Sciences in UCSI University of Malaysia. Obtained Bachelor of Pharmacy from the University of Malaya, PhD in Pharmacy from the University of Nottingham. She has worked as a hospital pharmacist, industrial pharmacist, and community pharmacist before joining academia at UCSI University, Malaysia. Dr. Mogana got many awards and recognitions. Gold Award at the Malaysia Technology Expo, Venus International Women Award, Associations of British Inventors and Innovators Award, Silver Award at the Inventions, Innovations and Technology Exhibitions, HBO Vice Recognitions, editorial board member of the Asian Journal of Pharmacognosy, scientific chair of the National Pharmacist Conventions. The areas of interest in her research is about antimicrobial natural products research to address antibiotic resistance, natural products derived polyharbor formulation for women disorder and related cancer, natural product drug discovery, dual inhibitions of pain or inflammatory bioactive markers, natural product derives polyharbor formulation for skin disorder. She has published approximately 50 research articles. Most of them is about antibiotic, anti-inflammatory, anti-cancer, and formulations. Here, some of Dr. Mogana publication related cancer and anti-cancer. In vitro cytotoxic activity of medicinal araliaceae against KB cells, screening of tropical rainforest plants of Malaysia for in vitro anti-cancer activities, in vitro anti-tumor potential of Canarium patentinapium mix, anti-angiogenic effects of ethanolic extract and its phenolic-rich fractions of Acacia auriculiformis bark in the chick embryo chorioallantic membranes model, anti-angiogenic effects of ethanolic extract and its phenolic-rich fractions of Pellicium decipiens, the chick embryo chorioallantic membrane models. Explorations of biomarker of the diagnosis, treatment, and prognosis of cervical cancer. All right, ladies and gentlemen, before the presentation starts, let me to tell you how our presentation will go. First,
Okay. Can you hear me? All participants, can you hear me? Okay. Yes. Before the presentation will start, let me to tell you how our presentation will go. First, our speaker will give the presentations approximately for 1 hour. After that, the question and answer sessions followed by conclusions. All participants can ask the questions via Zoom chat. The questions can be given in Indonesian or English. Ladies and gentlemen, here we come to the main event. Let's welcome our speaker, Associate Professor Dr. Mogana Sundari Rajagopal. Dr. Mogana, please be welcome.
Thank you, Dr. Zaria. Am I audible?
Okay.
Can you hear me?
Yes, I can hear you clearly.
All right. Would you like to share the slide? All right. Very good morning to the director of the organization, to the dean of the faculty, to our host, and also the MC. It is my utmost pleasure to be here. Thank you very much for the invitation. It's also my pleasure to be able to share with you all my perspective and viewpoint on the theme of today's discussion. Thank you very much. I'll wait for the slide. Dr. Zaria, you're sharing the slide, right?
Okay, wait a minute, doctor, please. Bisa langsung aja. Dr. Mogana, would you like to share your slide directly? Because I think we have something wrong in our connection.
Can you see the slides?
Yes, doctor. We can see your slide right now.
Can't seem to move the slide. It seemed to be frozen.
Dr. Mogana, we can't see your slide.
Yeah, the slide seems to be frozen from my side. I'm not sure why.
Oh yeah, okay.
But you do have a copy of my slides, right?
Yes, but we can't to share from our device, doctor. Need a password to open that.
No, you can just put as a view only. Frozen from my side. Am I a host? I can't seem to share. Why is that?
Okay, doctor, we will try to share with our device because until now we can't because your slide need a password. Okay, we will try. Please wait a minute, doctor.
All right. Is it okay now, the slides?
Hello, Dr.
Can you
Mogana.
all see my slides? Is it okay? Hello? Can you all see the slides now?
You can start your presentation.
Okay, thank you. All right, very good morning again to everyone. So before we begin, let me just introduce myself. So thank you to Dr.
Hello,
Zahid, the kind.
Doctor.
Yes? Can you hear me? Hello, can you hear me?
Kedengar.
Can you hear me?
Ulangi suara dia kedengar aku tidak baca. Saya tidak dengar.
Hi, good morning. I can hear you. Can you hear me?
Yes.
Yes, you
Yes.
are.
Yes,
We can
Doctor.
hear you.
So shall I proceed? I'm not getting it.
Yes.
Can yeah? Okay, right. Okay, so thank you again for the kind introduction, Dr. Zaria. So before we begin, just a short introduction where I come from. So I actually come from UCSI University in Malaysia, neighboring Indonesia, right? So at the moment, we are ranked at 284 in the world, right? So next slide. So as for the field of pharmacy, I think we are hearing some echo over the other side. If it is muted, yeah. Okay, so for the field, I'm still hearing echo. the field of pharmacy, we are actually top 300, right? World record. So that is a privilege for us to be at the top 300. We have been running the pharmacy program. We have currently 10 different programs, as in the next slide, for the past 23 years right now. So we have moved on from having a bachelor's of pharmacy program in the next slide, moving on to having our master's program, our PhD program, as well as our new diploma programs, right? So again, within the faculty, in the next slide, we have some achievements that I feel that due to the hard work of the faculty members and researchers. So we have over 3 million ringgit worth of active grants, with a total 144 publication last year itself, with a majority of the papers being in Q1 and Q2 with an impact factor over 250. And then we also have 4 patents in 2022 with 5 copyrights. So I must say that all this is due to the hard work of the faculty members and the support from the university per se. Now, moving on to the topic of discussion today, which is exploration of medicinal plants as anti-cancer candidates. Next slide. So if we look at cancer per se, we look at the international statistics, and we see in the latest compiled statistics were up to 2020. So we can see the top 10 cancers in the world, right? Ranges from breast cancer, prostate, and lung, the top 3, until all the way down to thyroid. And we can see that the incidences are just very high, and the mortality is also nearly half of the number of incidences that's occurring worldwide. So this is quite alarming, because as we will see later, the number of drugs that has come forth, that has been in the pipeline for many, many years, it's not very encouraging, especially from the conventional drug discovery perspective. And however, we can see that the number of discoveries happening from natural product has been increasing. And why is that? Why are we moving on from conventional synthetic way until nature? We'll see shortly why. So next slide. And based on the top 10 highest cancers, we can also see that the number of cases worldwide, internationally, is about 19 million. And of this, also due to the population, 50% of them are from Asia, where we are now, Malaysia and Indonesia. So it's quite alarming that it doesn't seem to show a dip. It just keeps increasing and increasing. So we can say following Asia, there's Europe, and then there's Latin America, and moving forth to Africa and Oceania. Next slide. Now, if we look at with the 19 million cases, right, 10 million of that resulted in death. Now, that is definitely a very, very alarming statistics. And conventionally, if we look at what is being used for treatment, I think we all know that we have the chemotherapy, radiation, surgery, and so on and so forth. But what is the negative side of this therapy? Now, if you look at chemotherapy, which is being used right now, we use molecules that inhibits proliferative signaling pathways. It also induces apoptosis of tumor cells, and it usually consists of a single therapy or a combination of classical treatments, depending on the type of cancers and, of course, the biological conditions of the patient. Now, monotherapy or single therapy has been the traditional therapy in treating diseases because of the dogma that we have, one drug, one target conception. However, combined therapy definitely work much more efficiently than single drug-based treatments. Now, this is why, keep this in mind, this is one of the reasons why exploration of medicinal plant is superior because of this particular reason, the combination therapy. We'll see shortly later why. Now, even though anti-cancer research and drug discovery have been continuously increasing, the therapeutic arsenal, which is the chemotherapy, fails due to many adverse reaction, drug resistance, target specificity on most cases. Definitely, there is some success rate that is only in controlling the symptoms or minimizing the growth, but how much is the success rate? It's not there. Now, I mean, look at drug resistance on the other hand. If we look at how it happens, the most challenging aspect of chemotherapy because of several factors. For example, the population of the cancer cells, the tumor load, the host environment, and so on and so forth. So when a cancer drug resistance occurs, you need higher dose. So when we use higher dose in order to achieve similar anti-tumor effect as the initial dose, this definitely will increase our side effects. All right. So if we also look at the conventional chemotherapy drugs like cisplatin, methotrexate, these are associated with severe adverse reaction. All right, such as definitely the common ones, hair loss, GI problems, neurological dysfunction, bone marrow suppression, and so on and so forth. Now, this is where, again, medicinal product comes to save us. Now, next slide. Now, why are we looking at the nature? The slide before, I think. Yes. So if we look at this, mega diversity. Now, what is mega diversity? So moving on from chemotherapy, what we've discussed just now, mega diversity is a term that people use to describe different countries' biodiversity. That means the variation of natural products that they have. And natural means it can be from the environment, plants, medicinal plants, from the ocean, from the different kind of species of animals, and so on and so forth. Now, to qualify to be called as a mega diverse country, the particular country must have high level of endemism, that means locality, and the species must be very, very diverse. And they must have a marine ecosystem along its border. I mean, the definition is very well defined. So if we look at this slide, we can see that Indonesia and Malaysia is definitely one of the countries that has high mega diversity because of the tropical rainforests and the very, very old rainforests that we have. And now, why is this important? Now, if we look at the recent compiled data, out of 1,881 newly approved drugs, ranging from antibacterial, antifungal, antiviral, and so on, all right, 929 of them have natural origin, right? And they fall into the class of biological macromolecules, all right, or unaltered natural products or botanical drugs or derivative of this natural product. And from 929 having natural origin, another 952 are classified as synthetic drugs. The synthetic drugs also, however, have natural product pharmacophores or drugs that mimic a natural product. Now, from here, we can see that nearly 50% of the newly approved drug comes from nature. All right. Now, why is it so? Because nature stands as an infinite and inexhaustible resource for the development of novel compound, a scaffold, all right, to behave as an effective drug, chemotypes, or pharmacophores. So this is very, very important. Now, if we look at this current slide that's being on the slide, being shown now, we can see worldwide, when we consider all the countries that have mega diverse, Indonesia stands at number 2. Now, this is a very, very precious statistics because this shows that the global diversity, biodiversity index is so high in Indonesia between the number of plants, reptile, mammal, fish, and so on, that there is so much of treasures in the rainforest in Indonesia. So now the question is, how much has this been explored? How much has this been investigated? Is there even a need to investigate, right? So if we look at the next slide. So this slide we can see, this is how a typical rainforest, all right, deep in the rainforest will look like. Such a beautiful picture, isn't it? It gives you the calmness of mind, peace. At the same time, tons of treasures are inbuilt there. They're just waiting to be investigated, right? However, this also goes in the point that we may want to talk about how much of these rainforests are getting destroyed nowadays. How much are humans preserving so that we can actually use this to explore rather than getting rid of the rainforests, deforestation that is also happening rampantly. And when deforestation happens, it leads to extinction of all of these treasures that we have in our rainforest. So that is a point to ponder on. Now, moving on to the next slide. So now we see here, just to share with you a little bit of statistics from Malaysia, where I am from. So Malaysia rainforest, similar to Indonesia, is also among the oldest in the world, estimated at 150 million years of age. And more than 50% of the peninsula Malaysia is covered with tropical rainforests. And in 2001, Malaysia was listed as one of the 12 mega diversity centers in the world, including Indonesia as well. And of course, the number of species that we have, from animals to plant species, is very, very diverse. All right, but again, the question is how much exploration has been done when we compare to the rate of deforestation that's happening. Now, moving on to the next slide. All right, so now medicinal plants. So why are we talking about medicinal plants now? So we have chemotherapy. We have seen the negative aspects of chemotherapy. We have seen the extreme limitations of chemotherapy. All right, and this is where medicinal plants come in. We look beyond, we look to nature. Now we look at terrestrial plants or microbes or slime molds. These are important, very important sources of novel pharmacological active molecules. And they definitely provide a library of promising therapeutic agents. And despite the increased popularity of synthetic products, which has led to nearly 50% of drug nowadays, the secondary metabolites from plants remains vital for drug design. Why? Because their core structure serves as a template or scaffold, right, for the synthesis or semi-synthesis of novel substance, definitely for a specific disease that we want to treat. All right. So in cancer, if we look at it, the inexhaustible scaffold is there in the nature. So many things are not discovered. And where do we find, if you look at conventional drug discovery method, they have run out of scaffolds or templates for drugs. They need variety. Where do these variety come from? From the nature. All right. Now, now another thing to consider is chemotherapy, synthetic drugs. All right. The safety efficacy is also questionable. All right. However, naturally occurring molecules in the nature have been reported to have high patient tolerance and acceptance. All right. Now, if we look at next slide. Okay, here we can see that many studies have been going on for many years now, looking into all the different kinds of cancers and all the different kinds of phytochemicals and medicinal plants that has been investigated. Now, phytochemicals have been in the spotlight of cancer research since the very beginning. From the time that the first anti-neoplastic drugs have been discovered, like carcinophylline in 1954, vincristine in 1963, actinomycin in 1960, and so forth. All right. And studies related to these molecules have is still ongoing until today. And it's very important to mention that natural products not only serve as chemotherapeutic drugs, agents, they also function as chemopreventive agents, also as adjuvants in cancer treatment, where they work synergistically with other agents. And of course, the fourth and most important, they also behave as sensitizers in drug resistance. All right. Now, this is very important. If we look at the mechanism of how a particular drug works and why it doesn't work after some time, sensitization or the drug resistance is a main issue. All right. And if a particular agent can be a sensitizer to the resistance, all right, apoptosis definitely will increase. Right. Now, from 240 anti-tumor drug that has been approved last 40 years, only 29 are strictly synthetic drugs, all right, which is very, very low. Now, why is it? Why is it so? Because the benefits of these natural compounds have reduced adverse effects and the capacity to impact on multiple signaling pathways involved in the carcinogenesis process. This is where we can say that a particular, phytochemicals function on multiple pathways rather than just a single target. All right. And this is where also synergistic effects comes on. All right. And if you look in the past 10 years as well, synthetic compounds with pharmacophore of natural origin that mimic the natural product will also approved as anti-tumor drugs. All right. So it all comes from the nature. All right. So now if we look at next slide. Now here we can see. Now why are we looking at, if we click on it again, yeah, again. One more click. No, the next. Okay. So if we can, I think the previous slide. Okay. Now, if you look at this, medicinal plants. Now, there is a low risk of toxicity. Now, why do we say this? All right. Does it mean that only plants have low risk of toxicity? There's a reason why we say it, because they have been validated to be selective in their function. And most of the time when they've been approved for a certain cancer study, they act specifically on the tumor cells without affecting normal cell. Now, this is very, very important. This is what we call selectivity of action. All right. And this is also one of the indexes that we use in cancer studies. All right. To see that it is selective in its target and it doesn't affect the normal cells. Whereas if you look at chemotherapy, there is very, very low selectivity. And that's the reason why the side effects is immense, from hair loss, etc., because it also kills normal cells. The next thing is complementary therapy. All right. Medicinal plants have the potential to complement conventional cancer treatments or chemo to enhance the efficacy and reduce side effects. So what do we mean by this? For example, if we are using a certain chemotherapy that is resulting in the loss of hair loss, hair, or certain side effects related to this therapy, medicinal plants can be used in adjuvant or complementary therapy to address these particular symptoms that arise. Now, of course, then the third we look at is potential for combination therapy. So combination therapy is where we look at synergistic effect. And this is very, very widely used in natural or medicinal plant. When used as therapy, we look at either certain biomarkers being combined together. We look at certain extracts being combined together, especially what we call as polyherbal formulation, where there's more than one herb that is being used. All right, all these are potential for combination therapy. And why is this important? Because we look at improvement in treatment outcome. All right, when we look at traditional Chinese medicine, traditional Indian medicine, or even traditional Malay medicine, there's always a kind of combination that is used where we look at a particular agent reaching out to the target, another agent that minimizes the side effects caused by that first agent, and then another one that increases the bioavailability of the first agent, and so on and so forth. So this kind of combination therapy ensures that we get maximum outcome, right? And of course, the fourth one is a controversial point, which some may consider low cost, some may consider high cost. It depends on how we look at it. Now, if we look at low cost, we can say that it comes from the nature affordable, accessible, compared to synthetic drugs that we need to do it in the lab. However, we have to keep in mind also that we cannot continuously exhaust the nature, all right, by taking and not giving back. So sustainability of the nature is very important. So it will not be low cost at all times, because once we have a particular molecule from the nature, the question now is, how do we get more of this? One is we cut down more trees or medicinal plants, but then that will cause deforestation, and we don't want that. All right, we want to be sustainable. So then what do we do with this molecule? How do we make more of this? We can do it in the lab, synthetically duplicate this molecule, or there are certain ways that science has moved towards doing, so we will see it afterwards. All right, next slide. Okay, so if we look here now, in nature, when we look at nature, medicinal plants as per se, we can see from each plant comes tons of bioactive compounds. And these compounds are called as phytochemicals, and they range from polyphenols, flavonoids, alkaloids, terpenes, saponins, quinones, coumarines, whatever, the list goes on. Different kinds of chemical classification, we call it phytochemicals. All right, and these phytochemicals, they have a lot of various pharmacological actions in cancer especially. It can be anti-proliferative, can be cytotoxic, it can be apoptotic, it can provoke the cell cycle arrest, and so on and so forth. Different kind of, it works via different mechanism. All right, next slide. Now, having said all of that, right, if we look at, having said all of that, if we look at how do we explore, now we know this conventional treatment, we know the medicinal, where do medicinal plants stand in nature, we know that's very important right now. This is where we get our scaffolds, novel structures to be used as template. Now, what are the phases that involve in natural products research? So definitely the phase one will involve extraction of the plant, all right, and of course fractionation and so on, and or standardization. And what does this mean? It depends on the kind of investigation we are having. If we are looking into bioactive molecules, all right, we will definitely do extraction, fractionation, and isolation of that particular bioactive molecule. And the method we usually use is bioassay guided fractionation. All right. Now, if we are not looking at a single molecule, there are also studies going on extracts, fractions, all right, whereby they use the entire plant extract or fraction. And the drawback to this is that the amount of the quantity of the bioactive molecule in an extract or fraction may be less compared to a single compound, all right? But it does have its benefits, whereby it works synergistically. Many compounds are in that extract or fraction and they work together, perhaps. And now the third way of looking at it is what we call as polyherbal formulation. Either we look at a single herb formulation, or we look at a more than one herb we call as polyherbal. Now, again, why? Because of the synergism that we're looking at. Now, if we are looking at a single herb or we're looking at a polyherb, we definitely need to go and do standardization or fingerprinting of this preparation. So what do we mean by standardization and fingerprinting? So for example, if we take a vial of a herb, or a herb that, say the vial contains 3 herbs, for example. Now, how do I know that that particular vial contains that particular herb ABC? And what is the amount or percentage of ABC that's in that vial at all times? If I'm extracting something, I'm preparing this mixture at any time, any point in time, how do I know that the ratio and the amount and the biomarkers inside remain the same? So this is why we need to do standardization and fingerprinting of that particular mixture. All right, so that is very, very important, something that many people, many industries that actually come up with herbal formulations, that they commercialize the herbal products, do not do. So how much of the standardization happens in a single capsule is still questionable. All right, so after this, of course, we have in phase 1, the next part where we look at pre-formulation studies or formulation development. And of course, moving on that, we need to, hand in hand, sometimes it happens parallelly, we need to look at the pharmacological aspects of that particular herb or compound that we are looking at. And this can range in cancer studies, of course, looking at the various in vitro cell studies, and of course the preclinical that falls into the animal studies. All right, and nowadays, we even bypass the, we don't say we bypass, we do different kind of in vivo or animal study where it reduces the number of animals we need to use. And for example, we use pharmacoscintigraphy, where we use radionuclides, whereby the particular molecule is tagged to a radionuclide before being introduced to a particular animal, and usually it's rabbits. And then we can look at a lot of studies, especially in absorption, distribution, metabolism, excretion in the preclinical study that we don't get to do in normal in vivo animal studies. All right, so that's phase 2. And of course, once the animal study is completed and it's still viable, it's still active, the molecule, what we will definitely proceed to phase 2, which is a clinical study that has its own phase 1, 2, and 3. So this is the normal phases that we all know. Next slide. So when we look at the phase 2, the studies that we are concerned about, that is the pharmacological aspect of it, we start off with in vitro studies, definitely. That is the lab setting. We look at cell lines, tissue cultures, or whatever it may be. And then we expose the cell lines to a drug candidate or an extract or a herbal fraction, whichever it may be. And then, of course, after that, we estimate the cell viability using cytotoxic test. And then we calculate the cytotoxic parameters for each of the drug if there are more than one. All right. And of course, after that, we look at the selectivity index, whereby we introduce the drug in cell line, the affected cancer cell line, as well as a normal cell line. We compare the ratio and we calculate a selectivity index, SI, to look at how, what is the toxicity of the agent, the candidate that we're looking at, as opposed to cancer cell versus normal cell. So what we want is it to be more selective on the cancer cell and not affecting the normal cell. So this is what we normally look at, very, very basic. And the next thing we look at is, the next slide, we look at the in vitro, in vivo studies. And here comes the animal testing, where after, if it is efficacious, we then look at the bioavailability, the toxicity, the ADME, the pharmacokinetic parameters, and so on and so forth, before moving to the human studies. Next slide. Now, if we look at this particular slide here, this the top part talks about the traditional approach. When we talk about exploration of medicinal plants as anti-cancer agent, we need to look at what are the different kinds of exploration method that people have been using. Now, conventionally, or the traditional method is the top part where we look at ethnopharmacology data or ethnobotany data, and then we do trial and errors from there, and then we check the extract for any activity, and then moving on, we look at until we come to the isolation of the compounds. Now, what is ethnobotany? Now, if we look at ethnobotany, it is a study of complex relationship between any culture, all right, and the use of plants in that particular area. All right, for example, Malaysian culture, and what kind of plants we look at in our vicinity, similarly to Indonesia. So this focus primarily on how plants are used, managed across many societies. Now, on the other hand, if we look at ethnopharmacology, now this one is defined as interdisciplinary scientific exploration, all right, on indigenous drugs or biological active agents, all right, which means the first one looks at how cultures, ethnocultures look at botany or plants. The second one, ethnopharmacology, looks at how different cultures, all right, the information from these different cultures are being used interdisciplinary to check on the pharmacological activities for a certain disease that we're looking at, all right? So which means that the latter, which is ethnopharmacology, has a broad focus in exploring medicinal agents, all right? And this is the first step that we use to investigate any potential causal relationship between the compound and the disease, all right? And over many, many years, ethnopharmacology has significantly contributed to the field of indigenous traditional medical knowledge of a particular society or region, all right? So if you look at this particular slide here, all right, we can see that moving on, when science caught up and we have what we have now, computational chemistry. So computational chemistry is now being used when a particular compound that has been identified from medicinal plant is now looked into in the system using a particular software. They look at a particular anti-cancer target of how this candidate can potentially affect a particular protein target. And then if it does show some anti-cancer potential, then we verify that data with experimental work in the lab, all right? And this then to prove whether the particular use of this drug among different ethnicity or indigenous people is actually real, because sometimes in a particular society, they say that, okay, this plant is being used as an anti-cancer candidate. Is it true or not? So how do we validate this with scientific evidences? So we can actually perform what's on the slide here, the extraction, whatever not, and then use the compound, check it in the system. Does it really work? If it does, verify it with experimental validation, and then giving an evidence of yes, it works, or no, it does not work. So this is the traditional way, and we have improved. We have some improvisation of the computational chemistry over the years of investigation. Next slide. Now, if we look at this one, this is another way of putting the computational part in a clearer perspective. So we can see the bioactive compound at the top, the molecule from the medicinal plant. All right, and how do we get this? Of course, by running a basic cell assay with the extract, and then if it works, bioassay guided fractionation, if it works, and moving on to do the isolation of that particular compound until we derive with that molecule. Now, once we have that molecule, of course, we do the docking, molecular docking. And after looking at the docking, all right, we move on to verify with in vitro and in vivo studies until it goes to clinical trials. All right, now next slide. Okay, now if we look, sorry, the previous slide. Okay, now if we look at this, this is another perspective of exploring medicinal plant, and that is called bioprospecting. This is, of course, not a new technique, been used for many years now. Just to share here what has been done and what is going on. So bioprospect is where we look at not so much from only ethnopharmacological data. We could be looking at literature, databases, or anywhere. But what they do is they pick up molecules, all right, from medicinal plants that are bioactive against cancer cells. And then they do a high throughput screening of many molecules that they have identified, they have shortlisted, and they do this assessment and moving on to in vivo and human trial. So this prospecting is done on a larger scales, usually by pharma industries, to look at potential candidates that they want to use as it is, the molecule to be used as it is, or the molecule to be used as a scaffold to do synthesis or semi-synthesis, right? So this is another way of looking at it. Next slide. Okay, now I would like you all to just watch this particular video. Here we will see how, although medicinal plants are very important, responsibility in its usage is also important. So let's have a look at this video first. Can press the play button there. Thank you. The play button is at the bottom left. So if you move the, yeah. No volume, yeah. Okay, has the video stopped playing? I think it's not the end yet, right? Okay, all right. Okay, so actually I included the video so that, you know, we wake up a little bit with music, but I do not know, the music is playing fine on my slide, doesn't seem to play over that side. It's okay. All right, so from here we can see that, from the video we can see, moving to next slide. So we can see that sustainability is very important with whatever we take from the medicinal plants, all right? Otherwise, extinction is going to happen of many species, all right? So what scientists have been doing is what we call, they use systems biology, moving on to synthetic biology and metabolic engineering. So what is the meaning of all of this? So we look at, we have clearly understood that plants represent a seemingly an inexhaustible source of biological active compound. We know this, all right? Unfortunately, most of these phytochemicals that naturally accumulate in the plant exist in very small quantities as it is, all right? And their complex structures, all right, usually render total chemical synthesis highly impractical. It's very difficult to do total synthesis of these molecules because of the complex nature of these molecules. That's the beauty of nature. It gives us a wonderfully creative molecule that human beings cannot think of. And to duplicate this in the lab completely with total synthesis is very, very impractical, very difficult, time consuming. So what can we do? So many of these plant derived compounds may be produced semi-synthesis, via semi-synthesis. However, it's of course pricey, all right, very expensive. So what happened for the past 15 years? Synthetic biology started. What is synthetic biology? It is where we use engineered microbes for the practical production of cheaper anti-cancer drugs from plants. All right, so if we look at this particular figure here that's been adapted by one Trends in Cancer paper, we can see here is an example of how globally people have been using to produce this particular all these molecular compounds. So they engineer microbes from particular targeted from a particular precursor. All right. So this development process relies on combination of system biology, synthetic biology, and metabolic engineering strategies. All right. So how does it happen? Usually begins with the search of a biosynthetic gene in the medicinal plant. So once that has been identified, characterized, these genes are transferred, all right, into a microbial host. All right. How? By genetic transformation. All right. So then the improvement of metabolic flux and optimization of the fermentation strategy will be used to achieve or improve the production yield of this engineered microbes, whether as a precursor or a final product. So we can see here in this thing here, using system biology and synthetic biology, they will identify the gene from a plant that is responsible, all right, for the production of that compound, and that will be transferred to a microbial module, all right, usually with DNA assembly. And once that is done, all right, the enzyme engineering happens. They will balance the cofactors to have maximum output of that compound. And then you will have the optimization so that the particular precursor is there in a quantity that they want. So this is how all the drugs we can see here, vincristine, camptothecin, taxane, and so on and so forth have been produced on a larger scale, all right, using this particular exploration. Moving to next slide. Okay, so if we look here, now what is this? Now, this is the latest, we could call it, not so latest, couple of years already. This is the usage of systems of a different kind of exploration. We have seen 3 so far. This is the 4th one, where you use systems or network pharmacology. Now, we can see that although natural products with anti-cancer effect have been identified and isolated from the herbs, and so on and so forth, even using system biology, we have managed to produce the particular molecule. What is lacking is usually the pharmacokinetics criteria, the absorption, distribution, metabolism, and excretion of this drug. All right. And due to time-consuming and costly traditional methods, this is usually also an obstacle in exploring medicinal plant as an agent. All right. So what has happened is recent years, there has been a holistic approach using computer-based system approach that integrates large-scale experimental studies, all right, and computational analysis to provide a mechanistic understanding of how the drug, all right, what is drug's action across multiple scales of complexities from molecular to cellular to tissue and organism leads to optimized molecule. All right. So we develop, there is a development of a natural product repository with this systems method. So we can see here, right, whereby using systems or network pharmacology, all right, we use in silico docking method. There is also QSAR, quantitative structure activity relationship, and of course ADME. In terms of initial screening of the compound, whether it will give the expected outcome that we desire. So conventionally, of course, we have to do this one by one in the wet lab. All right, and it takes a lot of time, a lot of resources, can be very, very costly, and most importantly, very, very long. So by introduction of network pharmacology, we have the particular candidate introduced in a system, computer-based algorithm, where we check the interaction of this molecule at various levels, from body to organs to cell level to molecule level. How does it interact not only to one target, but to multiple levels? All right, that is the organ, tissue, cell, and molecular targets. So we look at how it interacts on a big system, and from this we derive the what you call the candidate. All right, which means that we are reducing the time span needed to have a molecule that is has less toxicity, high efficacy, and better bioavailability. All right, so this is one of the ways that researchers are looking at to improve the efficacy of the compound, the delivery system of the compound, by looking at the pharmacokinetic issues. All right, as well as it enables us to derive a suitable derivative, especially in terms of efficacy and bioavailability. All right, so this is one method that is widely used now. Moving on to the next slide. Now, this is an example of the same of a system network pharmacology that is already the portal is already up. This was published in Nature. So if you look at it, this is a cancer HSP system, they call it, whereby they got 2,439 anti-cancer herbal medicine with 3,000 over anti-cancer ingredient or molecule. And for each ingredient, the molecule structure, the 9 key ADME parameters were provided. So we look at the figure there, we can see the info of the molecule. All right, we can see the molecular nature, the molecular names, the cells that it works at, the target that it acts at, and the molecular structure of that particular compound. So why is this important? All this is then deposited in a database, and this will help to reveal the molecular mechanism of a particular anti-cancer agent. All right, and this definitely helps in drug development, especially if you want to use for new drug discovery or drug repositioning. That means using an existing drug, changing the usage of this drug, looking if a particular drug is used for A, disease A, can it be used for B or C or D? And this method became very, very popular, especially during COVID pandemic, where people were trying to discover the cure, and everybody was looking at, to begin with, drug repositioning. So they were looking at many of the existing antimicrobials, whether it can actually be used to hit on the targets of the COVID virus. All right, so this is one example. Moving on to next slide. Now, if we look at global efforts, now we've seen all the different kinds of exploration methods of medicinal plants to be chosen as an anti-cancer agent. But globally, what is happening, all right, in terms of using this active plant compounds? So if we look at NPB, or National Cancer Institute, they have a branch where their main duty is to be responsible for the acquisition of various natural product material, not only medicinal plants, but all kinds of natural product for extraction and screening in 60 different kinds of culture cell lines. And they have this information with them. And what kind of tests do they conduct? They do isolation, chemical identification of this compound, and then run it through in vitro, in vivo. And then if it is good, they scale up the production of this active compound for preclinical and early clinical evaluation. So globally, this is what's being done. Now, in terms of if this is being done globally, in the next slide, are there certain policies or strategies that condones this efforts? All right, next slide. Okay, so we look here, 2014 to 2023, exhausted this year. Traditional medicine strategy was there by WHO, where they recognize traditional and complementary medicine, including the use of medicinal plants for cancer prevention and treatment. Now, the strategy is there, but how much has it been implemented, executed? That is a question, all right, in many, many countries. All right, so I think even in Indonesia or Malaysia, we can look at our own setting. Our national policies are there in terms of medicinal plant research, but how much of it is being implemented in terms of, or being allowed in terms of usage as complementary medicine or even integrated medicine. So that, of course, there's a lot of gaps there that need to be identified by the policymakers, by the researchers, universities, and industries. Next slide. An example in Malaysia, okay, there has been many studies being done on this. Famous one is Kemenkes nutans, among others. There are many compared to this, yeah, where it has shown to be an effective and complementary treatment. However, again, the implementation and execution to bring this therapy in complementary or integrated therapy is something that really needs to be looked at upon. It is an area where there's a huge gap and so many things can be done, especially integrated and complementary medicine. Unfortunately, it's usually static in some countries. Initiative is there, strategies are there, but implementation, execution is not there. So usually industries and universities like us, we go through certain point of research, we get an outcome where, hey, this can be used for certain therapy. We have the evidence to shout about, but nothing gets done, it gets stuck. There's a bottleneck where nothing gets done from there because of the implementation and execution issues, right? Next slide. So in terms of a Malaysian example, just to share, initiatives to promote bioprospecting. That means looking at entire database and selecting molecules. So we have our Malaysian tropical forest, whereby there is a Malaysian biodiversity information system that has been launched to facilitate the access to information on biodiversity studies and management in Malaysia. All right. And we also, the government also collaborates with many international partners, especially UNDP, to establish conservation program that includes bioprospecting new drugs. Having said this, the execution and implementation, there is a lot of gap that can be improved on. Next slide. Now, one of the area of research that's also booming at this moment, where a lot of people are looking at, is now we have candidates that are very potent, for example, as anti-cancer agent. But of course, the most important is having such things so potent, but how do we get that to the target site in its best bioavailable form? All right, so one of the things that is very important, especially for pharmacists, is we look at the formulation, all right? The mixture of herbs, which is a polyherbal formulation, but what about the dosage form for the human consumption? So we look at, of course, there are many oral formulation, conventional oral formulation, capsules, tablets, and this is very much so looking at many commercialized products. Capsules and tablets are the most predominant ones. Sadly, not many carry fingerprinting of the biomarkers. Many are just capsulation of powders and things like that. And of course, certain medicinal agents or mixtures that have poor drug solubility, for example, permeability, stability in GI, cannot be capsulated in oral formulation, and we need to look at other innovative formulations. Then we also have topical formulations that people have been researching. For example, certain skin cancers or certain uterine cancers, in terms of localized ointments, patches, and things like that. Next slide. And of course, the more advanced methods like injectables, liposome-based formulations, and nanoparticle-based formulations, especially the gold nanoparticles. So many of these are coming up, and it's being rampant. And this is a challenge in terms of, for conventional pharmacists or researchers, dealing with a medicinal agent or anti-cancer agent from medicinal plant. It's not an easy, it's not as easy as a single molecular compound, because assuming that we are having a mixture, for example, a polyherbal formulation, let's say 3 herbs mixed together, in order for this mixture to be designed, a particular dosage form to be designed, whether it's a nanoparticle or lipo-based formulation, it's a very different way of looking at drug formulation, because we're not looking at a single compound, we're looking at 3 different kinds of herbs or standardized herbs here. So we need to look at the biomarkers concerned, how do we ensure that they are there in the formulation. So it's a lot of challenges for the formulation experts, but this is something that is increasing, and it needs to increase because not only a single molecular compound is important. If you look at, for synergistic effect, there has to be multiple compounds, and so this is another area of research that's going on. Next slide. So in summary, if we look at the potential of natural products, especially medicinal plants, in the discovery of new anti-cancer agent, all right, there are multiple pathways that particular researchers can explore, or as we have discussed or seen earlier. So we can definitely moving from the secondary metabolites, we can look at the pathways that they involve in cancer metabolism, their regulation in the pathways, the initiation phase of apoptosis or regulation by the plant metabolite, so on and so forth. Many different mechanisms. We can look at whether they are working on a single target or a multiple metabolic target, and when you can look at whether this particular target is involved with one biomarker or multiple biomarkers, and then whether this is sufficient, and how do we then replicate it in terms of large-scale production. All right, are we going to look at how are we going to look at the toxicity, how we're going to look at the biotransformation of the drugs in our system, how we're going to look at how this particular drug improves life quality, that means minimizing the side effects. And of course, in doing all of this, we have to utilize our research tools. This can be the database of medicinal plants that we have seen just now. We can look at the traditional uses, the ethnopharmacological approaches. We can look at the chemical structures of this natural product and look at designing or changing the structure, adding on certain things to the structure. We can look at the drug targets and the metabolic or signaling pathways. We can look at the drug interaction with the systems or network pharmacology. So there are many, many ways or research tools that are available. It just depends on what are the pathways It could be one or multiple pathways being used by a particular researcher to develop these anti-cancer agents. Next slide. This is the summary that was discussed just now. I think the slide wasn't moving. Okay, next slide. Okay, so now if we look at what's the conclusion of all that we have talked about just now and what is the future prospect? Of course, it's undeniable that if we are looking at, we definitely need new molecules, new scaffolds, or new templates for drug design, especially anti-cancer. All right, and there is no, it's undeniable that plants are the source. All right, why? Because of the biodiversity that we have throughout the world. All right, 12 countries that have been classified as mega diverse. All right, and it's also important to highlight that there are very different classes of secondary metabolites in the plants that are responsible for the anti-cancer activity, and they can work not in one but multiple pathways in a cancer treatment. And then the advancements of technologies such as nanoparticles and so on has been very, very important because especially in the administration of these anti-cancer compounds, to ensure there's the sustained drug release and also reduction of side effects. All right. Now, extensive research work must be carried out on potential phytochemicals or the polyherbal mixtures to evaluate their possible application, their toxicity, genotoxic profile against a wide range of cancers, especially using the in vivo and in vitro and in vivo methods. All right, so having said all of this, all right, we also need to remember that working with medicinal plants need not be through one method or through one way. It could be a single molecule, as we've discussed. It could be an extract, it could be a fraction, it could be polyherbal formulations, because there is no one rule to the entire treatment for cancer, and all this needs to be investigated. And dealing with medicinal plants, we have to also remember that sustainability of nature is very, very important. And this is where all the systems biology and metabolic engineering comes in, that we need to work in an interdisciplinary approach with researchers of different background. So we have a comprehensive team to develop things at different stages. With this, thank you very much for sparing some time to in listening to my sharing session. I'll end my talk. Thank you again to the director of the institute, to the dean and host, MC, and Dr. Zaria for the kind invitation. Thank you.
Thank you very much, Dr. Mogana, for your informative and interesting presentation. Your slide is very clear, and I think we can get many information from your presentation. All right. From your presentations, it showed that as a tropical country, Indonesia and Malaysia have many medicinal plants can be explored to be anti-cancer candidate. To get active compound and formulate is not easy and simple. Many things to do, and it's so complicated, but it doesn't mean impossible to do. It doesn't mean impossible to do. The interesting question is how much exploration have been done, especially in our country. Okay, Dr. Mogana and all participants, now we are in question and answer sessions. In our chat Zoom has 5 questions, Dr. Mogana. Okay, I share the questions. Okay, for your information, we have 3 prizes for the first 3 questions. So our committee can contact you if you get the prize. Okay. The first question is from Mr. Raymond Arif from Akbar Yamasi. The question is, the use of plants empirically show pharmacological activity, but when laboratory tests with pure extract or fractionation actually show weak potential. What is the solution to this?
Okay.
Second, okay.
You want me to answer one by one, Dr. Zaria?
Okay. Please answer,
Okay.
Dr. Mogana.
All right. So basically, what I understand, empiric means that it's by observation or experience, which is the ethnopharmacological data. All right. That means a particular group of people have been using it for many, many years to treat a particular disease. That's what we mean by empiric. So empirically shows pharmacological activity. I'm not sure the nature of the question, because empiric means it's by observation of the data from the community, right? So sometimes, of course, why we do the pharmacological activity in the lab is to prove, to give some sort of evidence whether the particular plant actually does have its efficacy in cancer, for example, right? And many times we do find yes, it does have activity, but it could be also toxic. That's one issue. And sometimes we do find yes, it does have activity, low toxicity, and that's what we want. That is more ideal, of course. So if sometimes we see there's no activity or very weak activity, it means that it is not ideal for it to be an anti-cancer agent on its own. So then we have to look at whether traditionally people have used just that one plant, or they have combined it with a couple of other plants, what we call polyherbal, all right, to give a synergistic effect. So these are the things that once we look at the data from the lab, we need to go back and relook at the way that the particular indigenous usage has been for that particular plant. Hope that answers the question.
Okay. Thank you, Dr. Morgana, for your answer. Next, the second question from Firmansa from Hospital Siloam. The question is, does Malaysia's state health insurance system guarantee to the cost of the chemotherapy treatment with natural drugs?
Okay. Thank you, Ms. Firmansyah. Okay, so basically, if as what I have shared just now, the strategy of the country moving towards integrated health system. Integrated health system means that a combination between conventional modern treatment with natural or traditional treatment is used together in a particular therapy. The strategy and the policies have been there. However, the implementation has not been so great. And as such, we have not moved towards the health insurance system of including both. Currently, it just looks at chemotherapy. So again, there's a gap between the strategy and the implementation. Thank you.
Okay, thank you, Doctor. The third question is from Nurul Intan Fatimah, our student in Poltekkes, Kemenkes, Pangkalpinang. Herbal plants as anti-cancer are rarely used in the medical world. This due to their bioavailability in large quantities. However, to overcome this, an extractions of the active anti-cancer metabolites contained in this plant be used as anti-cancer without the need to use large amounts of the herbal plants. Please, doctor.
Okay, thank you. Ms. Nurul, I think the question is referring to the fact that if we are using herbal plants as anti-cancer agents, the individual bioactive molecule may be available in low amounts, and as such, extraction of this contained in this plant without the need to use the large amounts of herbal plant. Don't really get the in question, not very clear, but what I understand is that looking at the amount, what we call biomass. So when we look at this, if we have a plant and we extract the single molecule, which is isolation of the bioactive molecule that it has anti-cancer properties. So we are isolating, all right, and then the next question is we need to look at how to duplicate this molecule. So if we are able to do it in the semi-synthesis way, or if we're able to do it in total synthesis way, fine. But many times, this can be costly if the molecule that we're looking at is very big. All right, so what they do, they use the system biology and the metabolic engineering using by detecting the genes in the plant that's responsible for this metabolite, putting it into a micro and ensuring that is being produced by that micro due with genetic transformation. That is one way. Now, if we are looking at herb, the plant itself as a whole, the amount, and we want to use the extract, the amount of that particular bioactive molecule definitely is lower because it's an extract, not a compound. So again, this is not conducive if the anti-cancer property comes from only that compound, and we cannot be using an extract. We only use a combination of herbal plants, which is polyherbal, if we are looking at synergistic activity, because sometimes the activity of the synergistic activity of a mixture will be much higher than a single molecule. So this is where some investigation needs to be done. Sometimes certain things work together compared to one single molecule. Hope that answers the question.
Okay, thank you for your answer, Dr. Mogana. The next question is from Rahmawati Pelani Juria from Universitas Indonesia. How was doctors' experience during research in third phase of clinical studies to get natural product as anti-cancer? What are the obstacles and challenges, and how does doctor overcome this problem?
Okay, this third phase 3 of clinical studies means human trial. Is that what the question is asking? I think, not sure. I think it's referring to phase 3 of clinical trial, human trial. Now, phase 3 of human trial, I think that's a bit, I think very, very few companies in the world have been doing phase 3 for medicinal plant derived agents. Phase 1 and 2 is there. Of course, when it comes to human trial, the biggest obstacle would be legal issues, getting the ethical approval for the human studies. And that's where we really need to ensure that our preclinical data, it's very comprehensive, it's very detailed, and every nook and corner of questionable areas are attended to, that there's an answer for all those questions before we proceed to the human trial. Hope that answers the question.
Okay. Thank you, Dr. Mogana. The next question is from Maulana Malik Ibrahim from Siloam Hospitals. What are the most potential natural chemical compound as anti-cancer that will soon be submitted to NPRI and have the potential to become cancer drug that will be marketed globally?
Okay, thank you, Ms. Mr. Maulana. So I think this is a very ambitious question. We all want to know that. as anti-cancer agents that submitted to MPRA. Well, we would not know the actual molecules or formulation that has been submitted to MPRA because of it's not declared to the mass public. However, the limitation of natural product always comes in terms of registration. For example, in Malaysia, if we want to register a particular herbal product for cancer, it's not allowed by law unless with certain approvals, right? So because there are certain diseases in our Medicine Sales and Advertisement Acts that's not allowed to be advertised. So when what industries normally do, they proceed to register a particular herbal product as herb. So without specific claim, the claims will usually state natural wellness or general well-being, but they have evidence or papers published that it can be used for cancer. So that's one way. The second way is if they want to claim, or we call it high-end claim of a herbal product, there has to be accompanying preclinical and some clinical data. So the particular industries or researchers would need to provide this evidence to NPRA to actually have a high-end claim. And this is the two ways of looking at herbal products. But if the particular person wants to register a bioactive molecule from a plant as a new chemical entity, now this has to go through the entire stringent measure of moving on from the process of molecule to clinical trial. The whole range of studies need to be there to be able to register a bioactive molecule as a new chemical entity, just like any of our conventional treatment or drugs. So that is the normal way. Now, because of this, some of this, we could say there are pros and cons towards all three methods, all right? And sometimes countries will have different regulations on registering herbal drugs, all right? But what I've just mentioned is more towards the Malaysian settings, all right? And of course, only if the policy becomes a national policy that herbs can be used or integrated with conventional medicine, all right, then we would see more and more, probably more and more potential registration happening at NPRA's side with actual claim. Rather than what's happening now, it's all on the general claim, because otherwise they would have to spend millions to have a high-end claim. So hope that clarifies the question.
Okay, thank you, Doctor. The next questions from our lecturer, from Zulpiawan. The question is, lung cancer is the #1 of type of cancer that causes the most death in Indonesia. Can lung cancer be cured? And what plant treatment is most widely used in curing a cancer?
Okay, I think lung cancer is also the second in the world or third in the world as in the statistics that we have shared just now. Of course, it's very, very critical, and usually it's discovered too late to be treated as well. And can it be cured? Of course, if we look at conventional treatment, no. If we look at whether you catch it early, probably. All right, so then that's how it goes. Now, what plant treatment is most widely used in curing cancer? Again, cure is not there yet. It's still in studies. As shared in the slide earlier, we can also check out in all the databases that we have, what are the candidates that are currently being tested for lung cancer. And I must say that since pandemic, probably from 2021 after pandemic it's been in the recovery stage, more and more studies have cropped up, especially in terms of lung cancer, all right? And probably because a lot of drug repositioning studies have been conducted during the pandemic using bioinformatic techniques, where we look at existing molecule, bioactive molecule, and looking at how the design can be changed after the docking to look at whether it can now be used to target a different protein responsible for certain cancers. So yes, they are there, and we can actually get the data. Quite a number is there globally in terms of which stage of study it's at, whether it's still preliminary, at in vitro or in vivo or actual human trials. Hope that answers the question.
Okay, thank you. The next questions, Doctor, from Baharudin Togatoro from Stickers Arjuna Laguboti. In vitro tests, in vitro tests, there have been many effective anti-cancer drugs, but why in vivo tests, these drugs are not as effective as in vitro tests? What's the problem? Okay, please, Doctor.
Okay, thank you for the question, Mr. Bahardi. Okay, so I would say that, we have seen many studies published worldwide that many candidates are potent as anti-cancer. Now, again, as we discussed just now, potency as an anti-cancer is not enough. We need to investigate whether it is toxic to normal cells or it's only selective to the cancer cell. It could be, for example, most tannins are anti-cancer, has anti-cancer. You do a normal anti-S or anti-TSA, most tannins will give good potent activity. But then when we go and look at the toxicity level, it's quite toxic to normal cells. Then that's not a candidate that we are looking at, because we are looking at a candidate that needs to be selective, not affecting or minimal effect to the normal cells, but more on the targeted cancer cells. That's important. Now, how many of the researchers have filtered this way? They have to look at their own studies. Are we looking at this? Because if we look at just potency of anti-cancer test, anti-cancer activity, a lot of agents will be there, because most tannins, like I said, are toxic to cells, and they will kill the anti-cancer cell, but they should not be killing the normal cell. So that's point number one. Point number two is that the way that an in vivo test is being done will determine whether this activity is replicated in a living body. For example, the choice of the model, the animal model that we choose, is it specific to a particular cancer? Because we have multiple animal models for different types of cancers, and some of it we have a very limited way of viewing. For example, we take a xenograft model of a cancer, then we introduce that anti-cancer agent, and we're looking at whether the cancer that we have embedded is shrinking, or the kind of histopathological parameters that we're looking at. But what happens in the body of a particular rat or animal, for example, we don't know much. It's very minimal. So that's how pharmacoscintigraphy study comes in now. It's very useful, whereby we can view by tagging the molecule, the anti-cancer molecule, with radionuclide, introducing inside into the animal. We can actually see the molecule's absorption, distribution, metabolism, excretion. So from here, we can actually see, is it firstly being absorbed? At many times in vitro, it might be good, but when it goes into the body, absorption might be a problem. It could be a first pass metabolism issue. All right. Second thing, is it distributed where we want it to be distributed, targeted, or it's all over the place? So if that is the case, then we know that it is the formulation that we need to look at. Third thing is the metabolism. Where is it being metabolized? The fourth is excretion. So all this we will know using pharmacoscintigraphy studies, which we will not know with the conventional in vivo animal model. Then we think that, oh, it's not working, the histopathological size of the cancer is not so useful, so it's not working. But actually, there could be other issues happening in the body of the animal, that it's not because of the efficacy of the molecule, but it's because of the limitation of the ADME parameters. Then if that is the case, then we know, okay, then when we introduce it into the animal, we need to formulate into a dosage form that is has good absorption, distribution, and so on and so forth, so that the activity can be seen. So these are the things that usually happens, that unfortunately, that people stop at in vivo because they don't realize that many other things could be happening in the animal's body, and it's just a measure or it's just a matter of probably absorption and distribution. Hope that answers the question.
Okay. Thank you, Dr. Mogana. The next questions, Doctor, from Repi Mariska, our student in Poltekkes Kemenkes Pangkalpinang. In several studies, many say that there are some herbal plants that have a greater ability to suppress cancer of cancer cells than the standard cancer drugs commonly used. But why are these herbal plants still rarely used in medical procedure, even though there have been many studies that support the potential of herbal plants as anti-cancer? Please, Doctor.
Okay, thank you, Ms. Refi. Right. Again, it goes back to, unfortunately, the issues that most of us researchers like us in the universities are facing, because we do say that we do find a lot of instances where we find a medicinal agent that is good in suppressing the cancer or killing the cancer cells better than the standard cancer drugs. But why we don't use it? Because of policies. So in terms of research, needs to be really backed up by national policies of a particular country. So if the policy just stops at research and doesn't move on to commercialization or actual utilization of this therapy in treatment, for example, what we talked about, integrated health system, and then moving on to including and using policies to justify that, to have insurance companies to include that, nothing gets done. So the researchers are stuck with just having the research at that point, cannot use it because policies don't support moving further or beyond. So that's an important thing I think the governments need to look at. Thank you.
Okay, thank you. Next questions is from Dita Ramadini, our student in Poltekkes Kemenkes Pangkalpinang. The question is, what strategies are used for the development of anti-cancer medicinal plants cultivations, and do anti-cancer medicinal plants have side effects?
I think these strategies, thank you. I think the strategies that we use for development of anti-cancer medicinal plant cultivation, it's more, it depends on what are the agents that we're looking at. So if we are looking at a single bioactive molecule, one molecule that can be replicated in the lab, semi-synthesis or total synthetic way, all right, then there's nothing much on the plant cultivation that we need to look at because it can be done in the lab. Because we just need the scaffold or the template from the nature. But if we're looking at polyherbal formulations or single herb formulation, that this is where the plant cultivation comes about, and this is very much needs to be aligned to the agricultural policies of a country, how we're going to cultivate this plant, because when we're looking at a single herb or polyherb, standardization is very important, as we mentioned earlier, which means that the particular herb that we're looking at needs to be cultivated at a certain climate, at a certain temperature, and be harvested at a particular time of the year when the phytochemicals are at the highest. So all this needs to be taken into consideration, and this is where standardization comes in from A to Z. So that's plant cultivation. And second question, do anti-cancer medicinal plants have side effects? I think we would know that nearly everything under the sun will have some kind of side effects, even agents from medicinal plant or single or synthetic ones, right? But the side effect from plant has been shown statistically to be lower, especially for those medicinal plant that has been used for many years, for centuries, because it's been used for many years, tolerability is high, side effect is low. For example, let's just take our simple spices that we use in cooking, for example. We use for many years with low toxicity levels. All right. But having said this, we also need to bear in mind that when we're looking at medicinal plants or things that a novel compound, for example, or a plant that has not been explored, we suddenly have found that it has good anti-cancer properties, it can have side effects. All right. And this is where we need to see whether it can be combined with other plants in a polyherbal, for example, whereby in traditional Chinese medicine, Ayurveda, and all of those, or even homeopathy, Unani medicine. Unani is the ancient Islamic method. So what they do is they look at if one agent is giving anti-cancer potential, but it's also giving you some side effect, can we introduce another herb inside, whereby this herb will settle the side effects caused by this herb. When we combine both, the human being does not see so much of the side effects, but we see the anti-cancer activity happening. So this is the way that we work with plus and minus the formulation in the traditional medicine. Hope that answers the question.
Okay, thank you, Dr. Mogana, for your answer. May I ask you for some questions, Dr. Mogana?
Yes.
I have two questions. I didn't see the Spatholobus species in your medicinal plants in your slide. Have you experienced about Spatholobus, doctor? I hear Spatholobus its claim have a superior anti-cancer activity. In here we call bajaka. I don't know in Malaysia. And then the second question is, does every medicinal plants that have anti-cancer activity definitely have antioxidant activity? Okay, please,
Okay.
doctor.
Okay, thank you, Dr. Zaria. Are you referring to the bajaka species? Which species?
Spatholobus, yes.
Is it the bajaka species?
Bajaka, we call here bajaka Spatholobus purigenius.
Okay, because bajaka species usually they refer to three herbs, and there's three herbs could be the Camber, Uncaria, and Salacia species. That is one thing there is. I think I did not include the potential or the anti-cancer agents from medicinal plant in detail because the title of what we want to share today is the exploration methods. And if we look at like what I was saying just now, if we look at literature, many tons of studies has been done on in vitro, many all around the world. But then as we go into in vivo, it reduces and it reduces. But a lot of times, there is a lot of investigation in the beginning, but then when we look at whether there has been a continuation, there is none. There's no further studies. Why or not, we don't know. All right, but it could be because of all the limitations that we have just discovered. You know, people see very good in vitro when they go in vivo, it doesn't work because of the limitation. It doesn't mean that the agent is not working. It could be because of the limitation of the animal model, for example. So that's where science comes in with technology and all. We need to utilize these things. So that's why I did not put in a particular, because even in Malaysia, we do have certain potential anti-cancer agents from even, you know, mangosteen, we have seen Garcinia, we have seen Clinanthus, many species specific to a particular region we have seen, but it's usually it stops at a particular point where there's no continuation, probably because of the policy. So that's why I've not put it in the slides per se. Maybe that could be for another discussion, because I think if you're referring to Indonesia, I think it's more on the husk or the Litoris Spatobolus litoris or something, right? And this one,
Yes.
highly antioxidant. I think it's also used for weight loss, if I'm not mistaken. And it depends on the region. What are the things specific? That's what I'm saying, that Indonesia and Malaysia, for example, we are mega biodiverse in the world. But how much of these treasures we have used for our research that's very, very questionable. I think that researchers in the universities and the government needs to re-look at how can we explore this. There has to be certain fundings, there has to be some kind of strategies moving forward to explore these treasures that we have, right? And on your second question, whether anti-cancer agents can be antioxidant, most of the time, yes, some of the times, no. But are anti-cancer agents, what we have seen, alignment, antimicrobial agents are usually anti-cancer. They have the properties of killing. And the most important thing that we need to look at is this, the toxicity level. Because when it's killing a cell, all right, it could be microbial in nature, or it could be a living cell. When it's killing a cell, we need to look at whether it's toxic. Because if it is toxic, then that's a no-no for that. But if it is not toxic, then we move on. On whether it's antioxidant, yes, we have seen alignment of some anti-cancer agents also having some high antioxidant effects. Antioxidant effect we're looking at, we need to look at the two different pathways. That is the non-enzymatic method or the enzymatic method, because a lot of people will just investigate a molecule on DPPH, for example, or ABTS. Well, this is a non-enzymatic method. And they will get activity and they'll be very happy. But when you actually introduce it into a normal biological environment, we need to look at the enzymatic method, that is the SOD, superoxidase dismutase like activity that causes oxidative, that reduces oxidative stress and these kind of things. So if it has that kind of properties, means that it's good, because an anti-cancer agent that's also antioxidative, especially in having SOD-like activity in the body, will definitely reduce oxidative stress. Hope that answers the question, Dr. Zaria.
Thank you very much for your answer, Dr. Mogana. Your answer and your presentation very clear. I hope we can understand for what you presented today. Ladies and gentlemen, finally, we come at the end of this webinar, and I will give summarize from what the speaker presented. Natural products and their derivatives contribute more than half of all clinically administered drugs. They possess a significant position in drug discovery for treatment of cancer and other infectious diseases. This clearly demonstrates that anti-neoplastic substances useful in effective management of human disease cure in plants. It is also known that nature is able to produce a wide variety of chemical entities of novel structure. Many of the new and novel compounds isolated from natural source might otherwise have never been discovered, especially those of considerable complexity requiring the development of methods for the creation of neuron system. Natural products appear to be a promising source for new types of compound with anti-cancer activity. I think that's all for today. Before closing this webinar, I would like to thank you to our great keynote speaker, Dr. Mogana Sundari Rajagopal. Thank you very much for your time and sharing. This is definitely very meaningful material. I hope we can collaborate in other opportunity. And for all participants, thank you very much for your active participations. Let's give applause to our speaker and all participants. All right. Next, I hand it over the event to the MC. Please, Mrs. Ratih Puspita.
Okay, thank you very much, Ms. Zaria, with such have such a great moderator today, and of course, such an interesting presentation and explanation from our speaker. Before closing, I would like to thank everyone that willingly attended to this webinar. I hope this webinar will be beneficial to all of us. Also, thank you very much to our speaker today, Dr. Mogana. Thank you, ma'am, for sharing with all participants in today's webinar. And to the entire team on duty at this event, thank you for your cooperation and hard work, ladies and gentlemen. Let us close this webinar together with lafaz hamdalah. Alhamdulillahirabbil alamin. It's been great and wonderful day with all of you. Mirati Puspita Kusuma Dewi Purba. See you again in the next